Thank you for the introduction, Dan. I really appreciate that. You kind of took all of my introduction away. I'm just kidding. But I really kind of just want to talk about herbaria and digitization efforts among herbaria, but more so with the emphasis of California Botanic Garden's efforts in digitization over, well, probably since the 90s. So, but more recently, some of the digitization efforts that we've been doing in the recent years. So let's see, try to advance this here. So I know there's some herbarium people in the crowd, but I also wanted to kind of speak to those people who are not familiar with herbaria or what an herbarium is. And essentially that is a collection of preserved plant specimens or fungal specimens and the accompanying collections data that's used for scientific study. And so what I have here is a specimen on the left in RSA, and then also a specimen on the an image of our specimens in the collection on the right. These are housed in steel herbarium cabinets. So herbaria document the occurrence of the world's biodiversity and provide a reference for identification, characterization, and also classification. As you can see here with this picture, specimens are organized in a system to permit easy retrieval, access, and also for long-term storage. In our collection, we have, they're organized according to the APG4 collection, the Angiosperm phylogeny group. But some herbaria, some have not ever changed their classification system. So some are based on the Angler and Prontal system or the Carlquist system. And when I came on to California Botanic Garden, it was mostly the Thorne classification system. So if you are really, after you get through my talk and you really want to know more about herbaria, I would strongly recommend Barbara Tier's book. She is the former director of the herbarium at New York Botanic Garden. And this is a wonderful account of the history of herbaria and herbarium use. So I strongly recommend it if you don't have it already. There are over 3,400 herbaria worldwide and very close to 400 million specimens housed in these herbaria. The largest herbarium is in Paris, the National Museum of Natural History, with 13 million specimens. And in North America, the New York Botanic Garden herbarium is the third largest herbarium in the world, with 7.5 million specimens. Herbarium specimens can consist of a whole plant or plant parts. They're pressed, they're dried, and they're mounted to a standard size sheet of archival paper. In North America and the US, that standard size is about 11 inches by 16 inches, but it's slightly longer in European collections. On the left here, I have a specimen of a Bonnie Templeton collection of fern. And this is unmounted, so specimens come in unmounted and then can get mounted onto archival paper. On the right, we have a specimen that has label information in the bottom right corner. But as you know, specimens can also be mosses, algae, fungi, lichens, and they can be stored in archival packets. They can also be seeds, fruits, wood specimens, pollen, specimens that are preserved in alcohol, microscope slides, and also DNA extractions. Herbarium specimens are an essential resource for many disciplines in the plant sciences. They're a valuable tool for assessing global biodiversity change and loss, and they're increasingly used in research investigating phenological change, which I'll give some examples of that in the talk, as well as ecological niche modeling approaches and used in invasive species and looking at their impact on biodiversity. And importantly, herbarium specimens help to inform conservation management decisions. So over the years, digitization of natural history collections and herbarium specimens in particular in this talk really have been changing the way that we use herbaria and how we conduct biodiversity research. So what do I mean when I'm saying digitization? Essentially, that is the process of converting information into a digital format. And so this would include all the data from when a specimen was collected, including the specimen itself, and any content that's generated during the history as a specimen, and putting that into a digital format. So here I have Sherwin Carlquist specimen on the left, and Rebecca, this is probably one of my most favorite Sherwin Carlquist specimens. Cardiamanes from New Zealand. And all of that information is transcribed and put into a database where it can be accessed and more readily used. So when a specimen is fully digitized, it means that it has been databased, imaged, and georeferenced. And then you'll note in the top left corner, we also have a barcode, and that's part of the digitizing process, because for us at RSA, all of our specimen images, when we're imaging specimens, they're named after the barcode. And when I'm talking about georeferencing, and if you're not familiar with that term, when a location for a collection, there's no latitude or longitude coordinates with that location, we will assign one based on the locality provided. So here I have in the red box latitude, longitude coordinates that were georeferenced by one of our graduate students. I wanted to give a couple of examples of how specimens have been used. The data has been used from the efforts of digitization, and these are actually relatively old now, but digitization of herbaria actually has been around for quite some time, and specimens are being used in this way. This is one example where Lori et al., researchers were using specimen data from the Consortium of California Herbaria to look at how distributions would change with respect to climate change. And so they looked at Camelbatia foliolosa and Abies magnifica variety shastensis, and using herbarium specimen data and also ecological niche modeling approaches, projected that these two species that have adjacent present ranges would move in opposite directions. So that's one way that we've been using specimen data. Another way that we've been using specimen data is looking at phenology. So Everill et al. Looked at leaf out dates for New England deciduous forests, and that was all based on herbarium specimen data. And what they found was that leaf out dates were correlated with annual variation in April temperatures. So increasing each degree Celsius in mean April temperature about 2.7 days earlier. What's been fueling a lot of the digitization efforts over the last 10 years or so is an NSF program called Advancing Digitization of Biodiversity Collections, shortened as ADBC. They went into their last year, 11 years worth of a program. And the The mission of this program has been to enhance and expand the national resource of digital data that documents the existing voucher collections for natural history collections, and to advance scientific knowledge by improving access to digitized information that resides in vouchered scientific collections in the US. Facilitating the digitization of natural history collections and associated data provides a baseline from which to further biodiversity research, and also to provide critical information about existing gaps in our knowledge about biodiversity. Within the ADBC program, there's two different kinds of networks, and I talk about this now just to give you a little bit of background, so that when I do talk about the TCNs and the pens that the RSA collection is involved with, you'll know what I'm talking about. So a thematic collections network is a network of institutions that has a strategy for digitizing collections in order to address a particular research theme or a biota of a region, so usually looking at some impact of climate change kind of theme. And a PEN, a partnership with an existing network, is our institutions that often join later to an existing TCN, usually because they have particular collections that can make significant contributions to that TCN. iDigBio, the integrated digitized bio collections, is the national resource for ADBC and serves as the coordinating center for the national digitization effort. They facilitate the dissemination and digitization of practices and workflows, and establish integration and interconnectivity among the data that's generated through these collection digitization projects, these TCNs. And they also promote the uses of natural history collections data by the scientific community and stakeholders through enhanced research and outreach activities. The goal of iDigBio has been to digitize 1 billion natural history collections. And if you go onto their portal, you'll see this number here, where we're at 132 million specimen records. That's constantly updating. So I guess we're still a little ways off from a billion, but they're getting there. So I just want to turn towards the CalBG herbarium and what our efforts have been. If you're not familiar with our herbarium, I'll just give you a little bit of background here. We're the third largest herbarium in California, as Dan had mentioned. We have over 1.25 million specimens, and we have the combined holdings of both the RSA and the Pomona College collections. Pre-pandemic, we were acquisitioning about 14,000 specimens a year. Right now, we're probably acquisitioning about 2,000 specimens a year. I'm trying to strategize ways in which we can expedite our acquisitioning strategies. We have probably the best representation of Southern California plants, but we are worldwide in scope. This just kind of gives you a quick summary of the digitization efforts that our herbarium has been involved in since probably the 90s, where we started with data transcription of California specimens. But we've received funding for all of these projects, and usually digitizing different parts of our projects with respect to that funding. The 4 that I want to talk about are the last projects that we've been involved with, which are the ADBC related projects. If you were to go onto the Consortium of California Herbaria portal, the CCH2, you were to look at our statistics, which are updating pretty much like daily. You would see that we have over 702,000 specimen records, and about 48% of those are georeferenced, and about a third of those specimen records are have images with them. So the first TCN that I'd like to talk about is the California Phenology TCN, and I'm sure that most of you are really quite aware of that, especially if you're associated with any one of these collections here on this map. This is a collaborative effort among 28 institutions. And what you see on the map here are black dots represent institutions that are from the original TCN, and red dots represent those that are from newly funded pens. The goal has been to digitize 1 million specimens from California and to score the phenological data of target taxa. Those target specimens for digitization are oldest specimens to establish a phenological baseline before the onset of anthropogenic climate change, the most diverse plant families in California to understand evolution of phenological shifts, and also taxa currently monitored by the U.S. National Phenology Network and the California Phenology Project, all of which render this digital data immediately applicable to ongoing research. So applications in research involved with this TCN. The TCN hopes to answer such questions as which species are most in danger of phenological mismatches, which habitats or vegetation types are most vulnerable to phenologically to changes in precipitation and temperature, where might mismatches occur between flowering plants and agricultural crops and their pollinators, and is pollinator specialization an evolutionary dead end. Phenological research in California has been accelerated by this TCN and their digitization efforts. The scored phenological status of probably over 1.4 million specimens to date. And CCH2 now has these trait plots for looking at phenological status of species based on the specimen data. So if you were to go onto the CCH2 portal, query a particular species, trait plots to look at the flowering times or from some particular plants. This here is an example from the California poppy. And so this is based on almost 2,000 herbarium specimens. I thought that I'd put in Allen's daisy here for fun, because it would be kind of neat to look at. But I realized that if you look at the bottom here, you'll see that there's only, and all four of them are ours, and 2 of them are duplicates that are at UCR. So this probably doesn't give you a lot of information about Allen's daisy and flowering times, but I thought it would be kind of fun to add here. There's been recently published research from the CAP team, from Katie Pearson and Jen Yost and others, that have come out with a paper in Madronio in the phenology issue. And they examined how long-term and interannual climate variation affects flowering dates of the California poppy, and whether the magnitude of phenological sensitivity to climate varies across the species range. Specimens were collected from what they found was that specimens collected from chronically warm or dry sites flowered early, and the flowering date was more sensitive to long-term mean temperature than long-term mean precipitation. However, independent of these effects, flowering date was sensitive to interannual variation in seasonal precipitation, and direction of the effect depended on the season in which the precipitation had occurred. So, for example, warmer than average springs in the year of the collection flowered 2.7 to 3.3 days earlier for every 1 degree Celsius increase in spring temperature relative to the long-term mean spring temperature. Our efforts to date, well, we were first tasked with essentially digitizing about 100,000 specimens and also databasing 20,000 new collections. And to date, we're very close to meeting our goals. And I suspect that when the grant ends in July, that we'll have probably surpassed those numbers. I also wanted to mention that RSA often helps out other herbaria, smaller herbaria, especially if we're able to secure funding from NSF to do that. And so that was the case with the Anza Borrego State Park collection and also Cal State University in San Bernardino. We digitized, we imaged their collections as well. Another TCN that we've been involved with is the Southern Rocky Mountain TCN. And the goal of this TCN is to build resources and research capacity to enable increased understanding of plant ecological niches in the high peaks and high plains of the Southern Rocky Mountain Range. There are 39 participating institutions, and the goal here is to digitize approximately 1.7 million specimens. The Rocky Mountain Range, maybe some of you have gone there and spent time there. It's a really beautiful place to be. It trends in a north-south direction over a broad latitudinal range. And because of this orientation, is thought to serve as an important migration corridor for alpine and montane plants. Additionally, the Rocky Mountain Range has been suggested as important in the for the rapid diversification of many Western North American genera. And as a consequence, these varied ecosystems and geological substrates support numerous endemics and/or narrowly adapted species. Digitization of herbarium specimens provides the means to further investigate questions of geographic distribution, patterns of diversification, and ecological niche evolution. And in particular, the digitization of the Southern Rocky Mountain plant specimens will enable researchers to study contemporary patterns of distribution and identify ancestral signatures of diversification and predict future environmental change, as well as help to mitigate negative impacts such as climate change or habitat alteration. Here on the right is the map that represents the Southern Rocky Mountain region that's targeted in the TCN, that's highlighted in blue. And in red are collection locations from Marcus E. Jones, who was a prominent botanist at the turn of the century. He collected prolifically throughout the Western US. And we happen to have his collection because he sold it to Pomona College so that he could retire. So this is a major contribution to this TCN. We came on later as a partnership with an existing network, and we are tasked to digitize 60,700 specimens. So far, we're close to getting all of the barcoding done. We've databased and imaged about half of those specimens, and we're about a third of the way there with our georeferencing. It's been a little bit of a challenge for us because all of the Southern Rocky Mountain plant specimens are actually located in North American folders. So when it doesn't make sense for us to pull out just those things. So we're actually going ahead and digitizing pretty much all of our North American collection. So I guess NSF is getting a double for their money. So it's been a challenge. This has been a little bit of a challenging project, but we've really digitized a lot. So I'm only showing numbers here that represent the Southern Rocky Mountain region, but this is probably, you could just double this because that's probably what we've done. The Endless Forms TCN, Endless Forms, Most Beautiful and Most Wonderful. The title actually comes from the last sentence from Darwin's Origin of Species. And this is, the lead institution is New York Botanic Garden. There's 15 participating institutions in this TCN. And the goal is to digitize more than 2 million specimens from 15 plant families that have unusual morphologies. And what I have here is a list of the families that we are digitizing. Essentially, the taxon selection is characterized by one or more of 3 types of continuous morphological adaptations, each of which has evolved multiple times throughout the plant kingdom. So succulents, epiphytism, which is hard to say, and carnivory or insectivory. They're all really kind of cool families. And so what we're hoping to do in digitizing these families is to test evolutionary and ecological hypotheses, which I'll talk a little bit about in a minute, examine trait evolution and development in these families, as well as taxon delimitation. The digitization can also help with national and regional floristic projects and also species distribution modeling to look at how climate change may influence how these plants adapt or migrate or go extinct with climate change. So one hypothesis that the TCN hopes to address is increased morphological specialization leads plants to be less evolutionary labile in the face of rapid global change, increasing risks of population declines and ultimately extinction. A second one, plant families that show clines in adaptive morphological structures from generalized to highly specialized offer insights into the pattern and process of stem, leaf, and flower development for comparative morphological genomic research. We came on as a pen, and we have the goal of digitizing a little more than 70,000 specimens. Several thousand are particularly noteworthy that I would like to highlight here. One of them being Dr. Thorne's collections. Thorne's botanical interests and his publications were broad and diverse, with notable contributions to our knowledge of the remarkable flora of Southern California. And he is best known for both nationally and internationally for his synoptic work to develop a classification system to accommodate all flowering plants. Thorne's synoptic work on flowering plants was supported by decades of field trips and active collecting, including some of the most botanically significant parts of the world. And if you've seen any of Dr. Thorne's collections, you know that his collection numbers are pretty high. They number more than 65,000, and I would guess that they probably even get close to 100,000 because he had so many that didn't even have a collection number. These have special significance because many are from areas in taxa that are poorly represented in US herbaria. Another noteworthy collection I wanted to highlight is Sherwin Carlquist, who many of you would probably know already that he passed away in December. He is best known for significant contributions in systematic phylogenetic and ecological plant anatomy and structure and studies of island plant diversity. And he published prolifically on all of these subjects. His publications number about 340 publications and numerous books. Because of his systematically based research goals, Carlquist collected voucher specimens from some of the most biodiverse and undercollected parts of the world, many of which are otherwise not well represented in herbaria. And he had actually told me one time that some specimens that he collected, he knew were already extirpated, like some species, which was kind of sad. Especially noteworthy for the Endless Forms project are voucher collections that he made on several families, such as Aceraceae, Drosseraceae, Nepenthaceae, and Orchidaceae. And with his colleague, Alan Lowry, he had described 9 new taxa of Drossera from Australia, and those type collections are held at the RSA Herbarium. And the last one that I wanted to mention is the Cactaceae, which is probably one of the strongest collections held in the RSA Herbarium, owing in large part to the important historic and contemporary cacti specialists that were either centered at the garden or affiliated with the garden, such as Lyman D. Benson, David Griffiths, Edward Palmer, Elmer Yale Dawson, and Edward Anderson, and also contemporary botanists such as J. Mark Porter and Mark Baker. I feel like with all these TCNs, we really kind of learned how to streamline. And so we're only in this project for about 1.5 years. And I just think we're kicking butt on this project because we're almost all through our imaging, although it looks like we're not, but we have imaged almost all of the families. We have a few families to go, and those are kind of big ones, like the Cactaceae I'm saving till last. But we've made really significant progress on our digitization efforts. And I show Lauren here. She started off as an intern and moved into a specimen digitizer position. And she alone has imaged over 52,000 specimens. So she's a powerhouse. I don't know where she came from, but she's amazing. And the last TCN that I just want to talk about is the most recently funded one that we have gotten. And so I don't have a lot to say in terms of what we've done on this project, but just want to give you a little bit of background. So Asia, being the largest continent on Earth, includes a diversity of topography and geology, including like the world's tallest mountains and lowest landscapes and habitats, ranging from Arctic tundra to tropical rainforests and mangroves to deserts. And the plants of this region are incredibly diverse, with more than 1/3 of the world's 350,000 plant species. But documentation of this diversity remains inaccessible, and research about it is difficult because most herbarium specimens of Asian plants have not been digitized. So this is a heat map generated from iDigBio, and that shows the distribution of 43 million plant specimens. And that you can see where Asia is light colored there. That really represents the overall specimen records in that part of the world are much, much fewer than anywhere else. There are 25 participating institutions. Harvard is the lead institution on this TCN, with the goal of digitizing 3 million specimens from 36 countries. This map shows the target area in light yellow that will be digitizing. And then the project will especially focus on digitizing specimens from the unique and critically endangered biodiversity hotspots of Southeast Asia in the Himalaya Hengduan region. And those are indicated in the dark yellow. And so these particular areas will receive much more detailed data capture. Some of the aims that the All Asia TCN hopes to accomplish is to apply novel digitization methods and machine-based learning to propel biodiversity discovery. And digitized collections will be aggregated with international partners such as the National Museum of Natural History in Paris, the Naturalists in the Netherlands, and the Chinese National Herbarium in Beijing to access over 11 million specimens from Asia. And this will provide critical baseline assessments for global change, to assess global change. And the last thing I just kind of want to highlight is both the staff and in particular student participation in all of these digitization projects. They're really critical to the success of these projects. And well, I haven't started the All Asia project, but with the other three, we have been able to support 15 undergraduate students as interns and 7 graduate students. And so I have Eli here, who is digitizing for the California Phenology project up in the upper left corner. And on the right, we have Christina Varnava, who is a graduate student and now is at Santa Barbara Botanic Garden as the living collections manager. And in the bottom left, I just wanted to make note about what we used to do before the pandemic. We used to get together, and the herbarium was a really active place, and it is still an active place, it's just different. But these are students from Cal Poly Pomona, and they're barcoding for the California Phenology project. And they would come and spend an hour and get extra credit as part of their coursework. And then they would learn about natural history collections and have a hands-on experience. And what I found was that most of these students had never been to the garden before, and almost all of them, maybe with the exception of a couple of them, had never set foot in a natural history collection before. So exposing students to collections and providing opportunities, barcoding event, because it is the next generation of botanists and scientists that will be caring for these collections for years to come. And if you want more information about any of these TCNs or anything about herbaria or digitization, I'm happy to share that. And I also wanted to note my project support for the 4 projects that have been recently funded. And I can take any questions at this time. And I can even stop sharing my screen.
Let's look in the chat real quickly. I saw a couple questions. Oh, looks like Joshua Durr answered that one for you. Any comments about geolocation privacy of specific sensitive taxa? Ron Vanderhoff is asking, what is your personal opinion about this topic, and where do you think it will eventually fall out?
Oh boy, that has been a topic that's been kind of going on and off in the consortium of California Herbaria. I think it's a really good issue to talk about, and I think that we can't come to a real consensus on that. At least in my opinion, not as a consortium. I think because some people want to make all the data available, and I get that, but I also think that we have to think about how we protect some of the data, particularly for plants that are really sensitive, such as Dudleya. I went ahead and redacted, as part of the All Asia TCN, I went ahead and redacted anything that was listed on the CITES Appendix 1 list, because I thought that those were really kind of important, at the very least, to list those. And I have been having conversations with Naomi Fraga, our conservation botanist, director of conservation, to potentially redact some of the Dudleya stuff that Dudleya collections that we've been digitizing.
Another question coming from Dennis is, are there any special issues about pressing and mounting aquatic specimens?
Oh, if you want to learn how to do that, that's probably one of my most favorite things to do, because my whole master's thesis focused on 2 peatlands in northern New England, and there was a lot of aquatics there. And so I would take herbarium paper out into the field and mount it right there on the paper. So if anybody ever wants a demonstration on how to do that, I'm happy to show people how to do that. But I have offered like demonstrations when I teach my aquatics of Southern California plants. I haven't taught it in, I don't know, maybe a year or so. Maybe a couple of years because of the pandemic, but there are special considerations, and floating the aquatics onto paper is probably the best way to do it in order to not have kind of a tangled mess.
Yeah, interesting. Good. Okay, Dan is asking, any political issues with the All Asia project, especially in the China area?
I am not familiar with any issues about that right now. I know that like we're working with our collaborators, and political issues have not come up as a concern as yet. It might. It's a good question, and I can't really answer that very well. I feel like the natural history collections community seems to work differently than the rest of the political, on a different level, I think. I don't know. That's not a very good answer, but.
Okay, also from Dan, he's asking about, is all material accepted, or are you kind of picky using known botanists, biologists? How do you kind of filter through what comes in?
Yeah, we do have criteria for what we accept. The specimens should be flowering in or fruiting and come with collections data. I mean, we should know where it's being collected from. We also want botanists to be submitting information about their permits, because we don't really want to be taking on specimens that have been not collected with any permissions. So that's really important. We do look at the quality of the specimens, and we scrutinize, but I don't pick, say that, I'm not going to pick you because of, I don't know. I don't have those kinds of judgments on particular collectors, but I do ask for quality specimens that are deposited.
Next question coming from Ron. How can observations from citizen scientists via platforms like iNaturalist and CalFlora work in conjunction with physical specimens to inform better science? Any thoughts?
Well, have you ever used the notes from nature? Because that is one way that there are many different projects on there that citizen scientists can actually help to database specimens. And I'm actually working on, we had just, imaged all of our Orchidaceae collections, and I'll be putting those on to notes from nature in order to have citizen scientists help us to digitize, to database those specimens. So it's kind of a fun way of like connecting with the collections. And also there's opportunities for the community to, talk about specimens, and they get feedback on their data entry and lots of encouragement. So I think it's a really great way of the community to connect with collections themselves. I know that lots of people are using notes from nature also as a tool. I haven't really personally been using it. And I haven't really been applying that in the herbarium per se, but probably notes from nature would be the way that I would kind of recruit citizen scientists.
Okay, I think we're getting close to the end, unless anyone, oh, Rebecca, okay. Does your job take you into the field to collect specimens, or do you get to travel through time and space, mostly in the herbarium proper?
Oh, Rebecca, that's a good question. I guess I do the latter, but also I'm starting some new research in Red Rock Canyon State Park to collect and look at actually kind of retracing Twisselman's steps, because he had actually put together an unpublished checklist of the park in 1970, and there's never been any published flora of that area. And so it's a pretty dynamic area where 3 floristic provinces come together and 3 geomorphic provinces come together. And so, and it's, well, I live most part of the time up in Ridgecrest, so it's just a half an hour away from my house. So I'm going to be spending some time over there. So both, doing both.
Okay, I think we are going to finish up here. I want to say real quickly again, hope to see everyone next month, February 17th, when we'll be talking mushrooms and fungi with Joan Schwartz. Otherwise, thank you, Mayor, for coming and talking to us. It was great to have Rebecca talking with you, and Stefan and Cedar Joshua, and I understand some people from out in the desert herbarium areas, all here, possibly others together. It's neat when we can get our subsection of interest groups together and talk and learn and bring a wider audience. And it looks like on the chat, we're getting a lot of outstanding thanks, thank you, great presentation, thank you. What a good time to finish up.
Thank you. I really appreciated being able to give you guys a talk tonight and to connect with you guys. You guys seem like a really great group.
We are, yes. Okay, then. I think we are finished with another good session. And we'll all see you later.
Much.
Goodbye.